Hi everybody. Today I'm going to talk about sex-specific involvement of GABA-related microRNA in resilience and vulnerability to stress. Up to 80% of people experience some kind of traumatic events in their lifetime. However, it is surprising that the majority do not develop psychiatric disorders such as post-traumatic stress disorders. We also know that men and women react differently to trauma exposure. For instance, stress in childhood may increase depression in women and drug use in men. However, the relationship between sex differences and trauma resilience and vulnerability are not yet fully understood, and this was the focus of our study. So first, we exposed male and female animals to stress in different developmental stages, and then we employed behavioral profiling analysis by classifying them to unaffected, which means resilient, and affected, which means vulnerable, according to anxiety-like behavior that they demonstrated. For instance, freezing time. So here you can see the behavioral profiling analysis, and we can see that in males, the percentage of affected animals, the animals that were exposed to both juvenile stress and adult stress were affected in higher percentage compared to all other groups. Whereas in female, the percentage of affected animals were higher and similar in both adult stress and juvenile stress plus adult stress groups compared to control group. So then we wanted to see if and how this individual response are expressed in the molecular levels in order to better understand the epigenetic mechanism involved in this phenomenon. So we decided to look into expression level of GABA-related microRNA, which we already know that are involved in stress response. So we focused on two GABA-related microRNA candidate, microRNA 33 and microRNA 144, and we compared the expression between affected and unaffected animals in different subregions of the hippocampus in the dentate gyrus versus CA1 regions in both dorsal and ventral parts of the hippocampus. So here we can see that in male rats, increased level of microRNA 144 were observed in the dorsal dentate gyrus and microRNA 33 in the ventral dentate gyrus in unaffected animals, whereas in female, we observed in affected animals an increased expression of microRNA 144 and microRNA 33 in the ventral CA1. So since we found upregulation of this microRNA in males in unaffected animals, and in female, we found upregulation of this microRNA in affected animals, we wanted to find out if we could manipulate these mechanisms. So in males, we inhibited microRNA 144 in the dorsal dentate gyrus and microRNA 33 in the ventral dentate gyrus. And by this, we wanted to interfere with resilience mechanism and increase vulnerability. And in female, we inhibited microRNA 33 and microRNA 144 in the ventral CA1. And by this, we wanted to increase overall resilience. So here you can see the behavioral profiling analysis following the inhibition, and we can see that in males, the percentage of affected animals significantly increased following the inhibition, whereas in female, the percentage of unaffected animals significantly increased following the inhibition of microRNA 33 and microRNA 144. So in conclusion, the behavioral profiling that we use allows us to differentiate between affected and unaffected animals. And following the behavioral profiling, we were also able to identify differences in the microRNA between affected and unaffected animals. Next, we found sex-dependent effect of the expression of GABA-related microRNA on stress response both before and after manipulation. So this novel understanding of sex-associated epigenetic involvement in the mechanism of stress-related disorders may improve gender-specific diagnosis and effective treatment. Thank you very much for listening, and for more information, you can send me emails. Thank you so much.
Wonderful and perfectly on time. Okay.
Yeah, I did 5 minutes. Or...
Yes, you did 5 minutes exactly.
Okay.
I will let Siri be the first to comment. Please go ahead.
Yeah, well, thank you very much. That was a
Thank
very
you.
succinct summary of what is presumably quite a lot of work. How long did it take you?
I didn't
The amount
understand.
of, the amounts of work in the presentation. Like, how long did it take you to collect the data?
This it took 4 years, around 4 years to collect all the data. And this my PhD work. So yes.
Mm-hmm.
So yeah.
And this is the main story from your PhD work, is that?
Yeah.
Mm-hmm.
I chose the main results because I have just 5 minutes. So yes. Yeah, I had 5 minutes.
So I wondered if it might be more helpful to present the data using a sort of more maybe traditional kind of bar chart approach, because I found that there were 8 of the pie charts on each page, and that was I think my working memory can only handle 4 or 3 or something. So as a way of like making it easier for people to see the relevant contrast.
Okay.
I also think that, there are lots of fashions in science, and my impression is that pie charts are not in fashion. I don't know what you guys think. It's sort of.
Yeah, I think it really depends. I always get this like jolt to the system when you talk to the mainstream media, where they definitely want everything into percentages and pie charts, because I think there's the assumption that it's accessible. And certainly, that's some of the feedback I also got when trying to present, my work in a flash talk for a ERC, or there's something that they think is so, implicitly easy to grasp from a pie. This much is, for X and this much is for Y.
Yeah.
But I definitely concur that, the idea of, when you're writing up a PhD, you're going to have multiple variables, several things that you're considering all at once. But when you're presenting it has to be much more simple. It needs to be binary at most. I can't even keep 4 things in mind, Siri. So I think you have to deal with twins, so you double everything. But I can't do that. I've now given the advice that you should just have 2 contrasting cases and let the reader take from those 2 cases the difference. We all love spot the difference. Our kids certainly do. Just give them 2 things and tell them to just implicitly, by putting them side by side, people are going to make the comparison themselves and extract out the most valuable thing. So yeah, I definitely was going to give exactly the same, just much simpler. Just focus on 2 things at a time at most. And as for pie charts, yeah, it's a tricky one.
Okay.
Yeah, I want to add, I honestly don't know the answer. But I think that, Maayan, your research is, if I understood it correctly, it's very translational and super important for these treatments of psychopathologies that have to do with stress, like PTSD.
Yeah, and also diagnosis, yes.
Yeah. And I think that because we only gave you 5 minutes, right? We didn't give you 15 minutes, in which I'm sure you would have put the information. But recently I had to do this exercise where I had to present quickly. And I needed to do it 20 times in front of peers until we got it right. And I think you can start with a big question, because your research does translate to a big question. Like, how can we better diagnose resilience to stress? Or how can we treat stress better? And then you kind of put a person in a mindset. It's just an option, but you do put a person in a mindset that gets that question going throughout your research, and people can connect the details of the biology to the bigger question that relates to your final conclusion.
Thank you. Okay.
Really,
this sounds
great.
Like
yes. That you talked about that, bad things happen to lots of people, and that it isn't sort of inevitable that you end up with a problem. I do think that is another kind of big question type opening. So I guess you could potentially also combine those if that's your opening, then you could use the treatment translation also as a wrap up at the end, right?
Yeah.
Yes. Maayan, did you want to ask us anything?
No, no. I
Feel
don't
no
think
pressure. It's just that we are here at your disposal, you see.
Results already published?
Yeah, I already published actually several months ago. It's in translational psychiatry.
Yeah.
So yeah.
Wonderful. It's a very good avenue.
Thank you.
Yeah, so another thing you could do to promote yourself, which isn't a bad word, add like a link to your paper.
Okay.
That people who are interested in learning more can just either take a photo of a barcode on the screen or have access to it. And they also know from your talk that these are already published. And
Yeah.
in a wonderful place.
Okay.
Yeah, this is overall. People usually use the last page as a sort of, it's kind of often if you're giving a physical talk, it's left on behind you. So I learned from Rochelle Ackley, who was here just before us, from her ERC application, because during the ERC interview, you have, very, very short time to present your work, and then but the final slide is typically on. So you put like all of the braggy stuff in there, your website, like she had essentially a whole summarized CV on the final page.
Okay.
Which I also did. I mean, you don't necessarily have to do that for everything, but I did. That is what PIs do. Well, we have a picture of the team. That is a PI way of bragging. We're like, this is how many people are in my lab. And then you get to like also sort of gracefully self-promote by talking about how wonderful the people in your lab are, which is definitely super braggy, but it sounds like you're just being a nice person.
Yeah, you're right. Yeah, actually, in my whole previous presentation, I put it in the end, the picture, yes, but I will do this, yeah.
Self-promotion is not a bad thing.
I literally went to a course in self-promotion once for women, and it was called Graceful Self-Promotion. And we just sat there, and there were a bunch of women joking about what is the course for men called? Just shameless self-promotion.
Then we should do the shameless part. I think Maya definitely add.
Maybe I have one question. Maybe is it possible?
Yeah.
Here I didn't explain about microRNA, and because I didn't have time.
Yeah.
So I just wanted to ask if it's something that was important, like if to mention about what is the microRNA, about I don't know, the epigenetic mechanism, or this may be just one I wanted to ask if it's necessary to explain.
I think,
or it depends who
this is a 5-minute talk, right? So for every explanation you add, you have less time for your results. So it is a fine balance. But I mean, people are, for short talks, they're always going to say you should add this and this and this and this, and they never tell you what you should take out. So I think what you could ask us is there anything in your talk that you could have taken out to be able to explain the role of microRNA? That's a really tricky question, though. I don't want you to ask me.
I think I can't do it yet, but there are some people who just, I'll use the word gracefully again, but they're just amazing at giving you just the right amount of introductory information. Again, using an example, and I'll go back to this binary idea that they allow you, and I take this from the Montessori education method, where they don't ever tell you things explicitly. They'll just be doing the thing, and then you're supposed to extract out the knowledge from doing the thing. They don't tell you this is how we make bread. You'll just come into the room and see the people doing all the different things, and you go, oh, that's how you make bread. And similarly, I think you can sort of lead people to understand some of the things you need to know. to make sense of the stuff that you actually need to explicitly detail. I mean, it's going to be very hard for you to just implicitly tell people, you know, about your finding, although that's what a picture is supposed to do, right? You show them an amazing graph, pie chart or otherwise, and they just go, oh, I understand what you did. Some things you're going to have to spell out, but I think the people who've impressed me the most, I remember I went to one of Lorimer Mosley's talks, Siri, and he was just like, he just would dive right in with a really poignant example, and you go, oh, I know what this talk is about. And then if there's a term that needs explaining, you take 2 seconds, but you've saved time because you've led them to understand some of the other things implicitly. So I think there is a way. I haven't got it yet, so I can't tell you how to do it. But there's something about leading people to knowledge without explicitly saying it, and therefore saving a lot of time.
Yeah, and let me just invite you to do something that we almost never do, which is, we told you this, now it's kind of vague, right? You're going to try and change your talk maybe. Just send us 2 slides, say this is what was before, this is it now after, is it better? And I guarantee you that 90% of the time people will answer you back, even very high level people. So I think that sometimes don't be afraid to do such a thing. Email us and we'll let you know if it's better. It was pretty good, right? We'll let you know if it's better than better.
Yeah.
Yeah. Okay, so thank you.
Thank you.
Thank you.
Thank you for listening. Thank you so much.